share. I can't share. It's not allowing me to screen share. But it's yeah. It's too soon. All right. Can you all see my screen? We're going to be covering biology of metastatic prostate cancer. And in today's lecture, we're going to be going on speaking about carcinogenesis, pathogenesis of progression, theory of metastases, biology of bony metastases, and if there's time, we'll cover chromosomal aberrations and candidate genes.
Mark, can I ask you to turn your camera off? It's just flashing a green screen at the moment.
No problem. Just a quick introduction. Prostate cancer, we know, is the most commonly diagnosed malignancy in older men. It's the most common cancer worldwide, the second most common cancer worldwide, according to the Global Cancer database. And up to 6% of men have distant metastases at the time of diagnosis. We have a 30% survival rate over 5 years for metastatic prostate cancer, and 9 out of 10 patients develop bone mets. At least one of these will develop skeletal-related events over a 2-year period if left untreated. To speak about carcinogenesis, we know from the Alford-Knudsen hypothesis that dominantly inherited predisposition to cancer entails a germline mutation, while tumorigenesis requires a second somatic mutation, whereas non-hereditary cancer of the same type requires the same 2 hits, but both are somatic. If we look at the pathogenesis of malignancy, we know that incipient cancer cells must overcome several hurdles to become malignant. It's a multi-step process that can take years or decades to develop, and at least 8 of these key attributes, which are listed on screen, must be acquired to move from a normal cell to a malignant one. Depending on the author, 60 to 70% of human prostate cancers arise in the peripheral zone, and the 25 to 30 in the transitional zone, and the rest elsewhere. On the cellular level, we've got 3 types of terminally differentiated epithelial cells that play a role: luminal cells, basal cells, and neuroendocrine cells. And then, although it's a debate which one is the origin, we know that they can develop from luminal or basal, and the luminal cells seem to, if the prostate cancer develops from luminal cells, seems to be more aggressive, but evidence is still conflicting, and we need further research on that. So although luminal cells and basal cells are generally the cells that cause cancer, we don't know about the neuroendocrine cells, if they can be transformed to develop prostate cancer as well. Some studies that they've done in mice and in rats have showed that certain oncogenes, when applied, such as the MYR80K1, can transform normal cells, basal or luminal. Once you've given the oncogenes, they can transform into prostate cancer cells, as shown on the diagram on the left-hand side. And when you add them to adenocarcinoma, or in the presence of castrate resistance, you can develop neuroendocrine as well. And then inactivation of tumor suppressor genes, such as your P10, your SMAD4, and your P53 in basal luminous cells, resulted in prostate cancer formation. To look at prostate intrathelial neoplasia, we think it's a precursor of adenocarcinoma, but not all cases progress to invasive disease. Diagnosis of PIN is made by luminal cell proliferation with dysplasia along the ducts. And when it does progress to localized advanced prostate cancer, it becomes locally invasive and then degrades through the basal cell layer and then through the basal lamina. How this happens, there's usually a loss of cellular adhesion, followed by increased motility, increased invasiveness of the primary tumor, entry into and then survival into the circulation, whereafter it enters new organs and eventually colonizes those organs. Shortfalls at any stage of this metastatic process could result of phenotypes of limited metastatic potential. So Gupta et al. Described specific genes that were tumorigenic. They were involved in initiation, progression, and virulence of these cancers, and they had to fulfill this cascade. The initiation genes gave the cells an advantage to enter the circulation. The progressive genes gave or fulfilled a rate-limiting step for colonization. And then the virulence genes provided an advantage to colonize secondary sites. And indolent versus aggressive cancers didn't have these genetic aberrations. And then if we look at Weisselbaum and Hellman, they noted that there was a paradigm. There may be primary tumor cells which have a limited capability in one or more of the necessary biological requirements for metastases. And they proposed then a biological explanation for the origin of oligometastatic disease. So once the cells have finally overcome the basal membrane and spread into the circulation, they need to once again overcome all of these signals to then become metastatic. So if we look at the METS theory, which has evolved over time, and the most recent one is our spectrum theory in 1994, and it was 1 year after its release was refined to describe the limited metastases of any solid tumor, and the term oligometastases was coined. The authors described two scenarios that both fell under the umbrella of metastatic disease, but likely have different clinical courses. The first are tumors early in the chain of progression, with metastases that are limited in number and location, and another group of patients with oligometastases who had widespread metastases, but then were mostly eradicated by systemic agents, where the chemotherapy failed to destroy the remaining tumor because of maybe the number, the drug resistance, or the tumors being in a privileged site anatomically. Currently, there's no definition on the consensus of oligometastatic disease, and I'll let my colleague Dr. Patel talk about that further. But generally, we have determined this definition by the number of detectable lesions on conventional advanced imaging, and the early retrospective data attempted to define oligometastatic cancer using bone lesions as the primary key inclusion. Emerging genetic data suggests that oligometastatic disease may be a unique biological and genetic state compared to widely metastatic disease. And while this isn't helpful with the definition, it's more based on the prognostic state of the patient's disease. So just to go through the biology of oligometastatic disease, it could be because metastatic growth potential is limited, and this may be secondary to environmental conditions in the primary, forestalling clonal pressure and spread. Cancer cells just do not slough out of the primary, or they do not have the properties necessary to survive outside in the circulation. They don't have the properties to invade, and they may land in areas that are inhospitable. Now, if we look at systemic disease, that's now the opposite. It could maybe be that the conditions in the primary are very poor, creating undifferentiated aggressive clones that spread, and they migrate out of the primary tumor. They have the necessary properties to survive outside and or invade target organs. Now, if we look at metastatic disease, which is a leading cause of death in cancer-associated disease, our conventional wisdom from Data et al. In 2010 suggests that lymph nodes adjacent to the primary tumor are often the first site of metastases, thereafter metastases to the liver, bones, and so on. Human prostate cancer bone metastases often present as osteoblastic lesions versus other types of bone metastases, which are often osteolytic features, and these metastases cause pain, hypoglycemia, and frequent fractures. Locally advanced prostate cancer metastasizes first to draining lymph nodes and then to the distant organs, but the hypothesis that this happens has also been criticized. Now, the 10-men study is from the hypothesis that each metastasis originates from a single tumor cell, where recent studies have demonstrated the existence of polyclonal seeding from interclonal cooperation between multiple subclones. So what this means is the cancer doesn't have to spread from the primary to a lymph node, then to the bone. It suggests that it can spread from primary straight to bone, bone to bone, and bone to lymph node. So the 10-men study showed in the context, and it was all androgen-deprived metastatic prostate cancers. They did whole genome sequences on early post-mortem studies, and they characterized multiple metastases arising from prostate tumors in 10 patients. They looked at the subclonal architecture of this metastatic spread, and they found metastatic to metastatic spread was very common. And transfer of the clones between metastatic sites was also very common. There was polyclonal seeding, which raised the possibility of interclonal cooperativity, remodeling of the bone niches by the initial colonizing prostate cancer clones, making it an attractive habitat for other clones to colonize later. And this has now also been reiterated by newer mouse and rat studies. So why are the bones important? So from an anatomical point of view, we know the vertebral venous plexus, in which the prostatic venous plexus immediately drains, is an important anatomical landmark, and it's an important role for metastases due to its location along the vertebral column. And based on the seed and soil hypothesis by Paget, which was one of our first metastatic theories, the growth of tumor foci tends to be in the as a direct result of a specific organ's microenvironment. So our prostate cancer, which are seeds in the bloodstream, settle in the appropriate soil, in which this case is our bones, or in our axial skeleton, which is red marrow abundant. From a molecular point of view, it becomes a bit more complex. We first and foremost is EMT, which is epithelial mesenchymal transition, and that's a way that prostate cancer cells needs to acquire EMT to be able to depart from their primary site and into the blood circulation. And then to invade bone sites, they need to have adhesion between the bone marrow endothelial cells and the tumor cells. And there we have a E-select ligand that plays an important role. There's also CD44, which is a multifunctional cell surface adhesion receptor that's also involved in as an adhesion molecule. The VCAM1, IGF1, and interleukin-17 are all able to boost this process of cell adhesion. After prostate cancer cells then have spread to the bone and then they nest there, they exploit the bone microenvironment. They compete with hematopoietic stem cells in order to occupy this little niche. And following this process, they become adapted to the bone microenvironment and exploit the mechanisms of the immune system and evade the immune system in order to survive. Once they're established in these niches, they enter a dormant state. And this has been called immune-mediated dormancy. And if we have high levels of P38, our cancer cells tend to remain in this dormant period. After they exit from this dormancy phase, they start what is called the vicious cycle of bone remodeling. This demonstrated on the diagram to your right. So the metastatic prostate cancer cells secrete factors like parathyroid hormone-related protein. These upregulate receptor activities of nuclear factor kappa B ligand, or RANK-L, and this expression by osteoblasts interacts with rank receptors on osteoclasts, causing bone resorption and osteoclastic differentiation. And this interaction continues, and it contributes to the release and activation of transforming growth factor beta, which is stored in mineralized bone matrix and further promotes tumor growth. So then you have more tumor growth and more parathyroid related hormone release, hence the vicious circle or vicious cycle. And there's also a DKK1 expression by these cells, which has a negative impact on this, and then MMP7 and solubilization of the RANK ligand in the bone increases bone resorption. So these are all the mechanisms in which bone metastases occur. Very, very complex molecular interaction. So then just quickly on ADT and castrate resistance. So we know that ADT inevitably results in castrate resistance through various mechanisms, including androgen receptor amplification, androgen receptor phosphorylation and bypass of the androgen receptor pathway, and increased androgen receptors in the cell. And then I will just touch briefly, because we're running out of time now, on the 3 categories of chromosomal changes that may play a role in the progression and development of prostate cancer. And they associated with genetic predisposition, which is our germline mutations, our BRCAs and HOXB13. Those are Q1 deletions generally. Then we've got our somatic alterations that result in amplification of oncogenes, your CMYC, your beta-catenin, and your telomerases. And then your somatic alterations that result in the loss of function of tumor suppressor genes, your P53 being the most important, P10 also well described. And then other metastatic tumor suppressor genes, as in your ecahedron and your Chi1, which have been shown to play a role in, especially in so yeah, that's essentially my talk in a nutshell. I'm not going to bore you going through this the rest of these the oncogenes, but just to summarize that they are important and they are an area of active investigation. And why is it important? We know because prostate cancer is such a huge burden of disease, and so many people with prostate cancer develop bone metastases and other metastases, that it's a high morbidity. And although we don't quite know the definition of metastasis yet, my colleague Dr. Patel will get there hopefully and improve our knowledge there. But the goal of this whole topic is to understand the additional targets that we can develop treatment options for in future. So like the P53 that's being tested as a potential drug to inhibit metastasis. Thank you.
Yeah, thanks a lot, Mark, for a great talk on such a complex topic. Do we have any questions on the floor or from anyone else? Okay, not seeing any. Okay, so we'll move on to a topic that might sort of be related to what Mark has spoken about in the biology of metastatic prostate cancer. So the second speaker is Dr. Bhavin Patel, who's a consultant Uro-Onc fellow, who will be discussing oligometastatic prostate cancer new insights. Thanks.
Hey, thanks, Sam, for the introduction. Just going to try and load up my slides. Give me a second.